today. Xing is giving the talk. So hello, everybody. And thank you for joining us today for the Molecular Connectivity Online series, or MCOS. I am Mariana Sala from the University of Liège. In case you are new here, the Molecular Connectivity Working Group is a group of researchers that aim to advance molecular imaging as a tool to study brain connectivity. We organize the online series as a platform to exchange ideas, promote scientific research on molecular connectivity. You can go to our website, subscribe to our newsletter, follow us on Twitter, LinkedIn to get the latest information. So I'm happy to introduce today's speaker, Dr. Xing, who is a research assistant professor in the Department of Biomedical Engineering at the New Jersey Institute of Technology. With a PhD in psychology, his research focuses on modeling brain connectivity using multimodal neuroimaging data, including fMRI, PET, structural MRI, and fNIRS. He also investigates brain alterations associated with autism spectrum disorder. He will give today a talk entitled Comparing Intra and Inter-Individual Correlational Brain Connectivity from Functional and Structural Neuroimaging Data. And the discussion will be moderated by Dr. Mattia Veronese, who is an associate professor in biomedical engineering at the University of Padua. Okay, so the floor is yours. If you have comments, you can write them in the chat, or during the discussion, you're free to ask your questions in the chat or directly. So thank you very much for giving the talk, and the floor is yours.
Thank you for the introduction. So today I'm going to talk about intra and inter-individual correlational brain connectivity from functional and structural neuroimaging data. So basically, the goal of my study is to study like brain connectivity or how different brain regions are related, or functional related, or structurally related. But using neuroimaging techniques, and we know that the most commonly used methods or simplest methods is to use some kind of correlations. Usually it is defined as functional connectivity or connectivity. I know that maybe there is some kind of disagreement, like whether you call it a connectivity. So usually I will refer to this as a correlation, simply as correlations. But occasionally, if I say it is a connectivity, bear me a little bit. So I know that so the study of functional connectivity started as early as 1980s. So those studies are using PET, usually it's a FDG PET, to study glucose metabolic activity. And those two seminal papers, like, you know, you see that they define like brain regions, like using this bondage, and then you see these regions, and then they can extract the signals that usually represent glucose metabolic rate, and then they can calculate some kind of correlations across subjects, and then you can generate a matrix, like they say the earliest matrix of like correlation matrices or connectivity matrices, like between different brain regions. And we know that those regions, we know they are working in a similar functions. They will show very high correlations, like in these two different separate studies. So because the measurement is the limitation of the measurement using PET, right? At that time, usually you only have one static PET image for one subject. So you have to do this kind of cross-subject correlations. But the most commonly used methods as later development, the first is functional MRI. They can measure indirectly the neural activity by using this hemodynamic response or blood oxygen level dependent signals measured using the both effects. Those effects are moment-to-moment fluctuations over time, right? It is so you can calculate these correlations within subjects. So these are one of the studies that I have done. So you see that those are 160 regions of interest that cover the whole brain. You see that kind of a rectangular, or this structure that along diagonal usually represent that those regions are from the same functional network. They show very high correlations. Those are within subjects. And of course, for PET, there's earliest development is also that if you use a constant infusion, right? This is also called a functional PET. They can constantly measure these fluctuations of the metabolic activity over time. If we can still do the kind of a correlation, these are within subject correlations. And within subject, and then you can still see that this kind of functional connectivity matrices that may be similar to the resting state functional connectivity. And basically there are two methods. One is the inter-subject, and another one is the intra-subject. And the problem for a lot of study from PET is that usually, although this functional PET is like this is the earliest development, but the most commonly used are still static PET. And usually it's one image per subject. So what we do is that, so for each individual, we have only one map, right? And then we have multiple individuals with stacking them together. And then you can see this as a kind of a time series, or we call it subject series. And then we can define regions, like two different regions. And those two regions, the signals will be somehow correlated, right? If in one subject, these two regions have higher metabolic activity, they will also have very high, the other region will also have very high activity. If another subject, the one region have lower activity, the other region is also tend to have lower activity. So then you will see some kind of correlations about that. And for MRI community, there is also similar measures, right? So for example, you can use brain volumes or cortical thickness, which reflect some kind of structural or anatomical properties of the brain. You can doing the similar thing that cross subjects, you can see correlations. But those are also called kind of sometimes they call it structural coherence or just like structural connectivity. And of course, usually when we talk about like brain communications, we talk about something that is like within subjects that do vary over time, right? Think about, for example, the functional MRI. Usually we have the signal going up and down, which we thought this indirectly reflect the neural activity up and down, right? Even though without any task in resting state, there are still some kind of fluctuation over time. And usually we want to say, okay, this reflects some kind of neural activity, and those neural activity fluctuations, if we see the correlations, it is more directly related to what we want to do. This is because the communication is really neuronal, right? Neuronal driven. But how does these two measures or variabilities correlated, right? This is the question. So just by measuring the outcomes, so for example, if we calculate the correlation matrices, right, we can calculate, for example, the third one is the functional MRI matrices that within subject correlations. And you can see that there are structure like the functional module-like structures. And also we have done this by using cross-subject correlation of FDG PET, right? You see this kind of a matrices, the whole brain matrices. And we also have this in brain structure MRI, we calculate the gray matter volumes. We also have this kind of matrices. And we can compare them to see how similar are they. So initially we saw this paper published in 2017. We see that there are some correlations, right? But you see that this correlation is quite limited. Here, I think it's very important to differentiate two things. First is that they are statistically significant. All of them are very significant in terms of statistics. That is because there are a large number of pairs of ROIs, right? Basically, each of the elements is one. Each of elements in this matrix represent one sample. So because the sample is very big, even though we only have a correlation of like 0.12, this correlation is very significant. But you can see that this correlation is kind of a effect size measure, right? That measure tells you how much is effect. One is the highest correlation, exactly the same, and 0 means that there's no correlation. And those correlations, even though it's significant, you see the correlation are not that high. And the highest correlation we found is between FDG PET and the fMRI. At that point, we thought like, okay, at least for the FDG PET, they reflect some kind of a functional relationships so that they are more correlated with functional MRI. On the other hand, the gray matter volumes, even though it is still correlated with fMRI, but that one have the lowest correlation. And of course, there are other groups like Eagle's group, they have used the other measures like structural connectivity measured using DTI. They also showed such kind of correlations, right? But they showed higher correlations, but most importantly, they show that this connectivity measures, functional connectivity measures, is more correlated with the structure matrices, if I remember it correctly, more correlated with the structure matrices than the functional connectivity matrices measured using resting state fMRI. That's what they found. So basically is that we want to say, okay, what drives these correlations, right? If there are two regions are correlated, what are the variabilities that give rise to these correlations? And the most likely that we want to have is like there are neural activity, right? And then the two regions have similar neural activity, even though maybe there is some kind of delays, but because of the slow hemodynamic response, they are showing very high similarity. So you correlated them, these two, they give you a very high correlations. And of course, for fMRI, there's other issues, confounding variables, such as physiological noises, head motions, and so on. But that's one we want to control them. But basically, we want to say that we want to interpret these correlations of functional MRI data as neural activity driven, right? That is what we want to do. But for intersubject correlation, whether we can attribute that to neural activity, that is the question. So for intersubject correlation, right? For example, like we can use metabolic glucose metabolic rates and measured by FDG PET. This is one image, and also we can have something like brain volume image, a segmented image from structural MRI. And so those kind of images, right? We are doing it across individuals, and we can still see that there are variability here. And how to interpret this variability, right? That is the question we are trying to. We know there are many factors, so that we are trying to tease them apart and try to illustrate like what kind of factors are they, how the different factors may contribute to this correlation structures. So in that sense, I try to do this, and I think there are certain different type of factors. I categorize this into these three categories. The first is kind of individual differences, right? For example, usually it's driven by, for example, the lifelong experience, right? For example, if you think about like brain structure, two regions are like always doing, you are always using these two regions more often, that those two regions may have very high volumes at the same time. And also maybe there are some genetic factors that determine these individual differences. And also there are some kind of a long-term effect for the aging, right? And this is very obvious for fMRI study, for MRI structural covariance study, because a lot of studies, there are available data sets that have a very wide age range, and usually they will attribute this kind of a covariance, structural covariance, to age effects. So for example, if they have a very wide age range from 20 to 80s, and then they can study the brain structure covariance, and which they also show that this covariance may be different between younger and older subjects. But this is kind of a long-term effects. But how this long-term effect may affect this correlation structures is the question that we have never shown that directly. And of course, so we most likely, to me, I think that we most likely to want to attribute this kind of correlations to this kind of state-like component, which is, I wanted to say, okay, if this reflects some kind of neural activity, right, that will be good. And of course, for PET, this PET data, we can say, okay, this is somehow indirectly reflects some neural activity, because the subject, when the subject was going in the scanner and trying to do the scan, and then they may think about something differently, right? It's similar to in resting state in fMRI scan. Sometimes they will think about like something like introspectively, or sometimes they just look at the environment and so on. Those may result in difference in metabolic rates in different regions, for example, the deformal network or the visual network, and then they result in a correlated structures across subjects. And of course, it reflects, for example, the mental states, like the mood and so on in the scan, during the scan. But of course, this kind of a state-like component can only be reflected in the functional data, right? For example, if we are studying metabolic rate, it may be related. But if we are studying structure data, it may not be, because I don't really believe that, although some maybe believe that there is long-term changes of structure, brain structures, that may be, but I don't think that, I don't feel like it is possible. So if we study structural brain correlations, maybe there's no state-like component here. So we have these different components. we try to tease apart different components, right? And then it's kind of difficult, but I think by carefully design, maybe we can at least show like different components, how different components may contribute to these correlation structures and give us some kind of insight. And later, if we have individual difference, we have this inter-individual correlation results can help us to understand or interpret results more better. So this is basically the goal of this study. So basically is that I think we can try to make a smart design to try to tease apart different part of these components. The first thing is that it's better that we have a longitudinal data so that we eliminate the individual differences, right? And then we only have within subjects, but we can still use the same kind of measurement we have, for example, the FDG pad over time, and also green matter volumes over time. And in that case, we can differentiate or minimize individual differences. And then we can comparing functional and anatomical data, right? Functional data, as I said, it may reflect this state-like activity. But structural data is not likely that reflect a state-like activity. So that if we compare these two, it gives us some sense, some idea about this state-like activity, how this can contribute to this correlation structures or functional connectivity. And lastly is that aging effects, right? We know that there are long-term effects. So we can model that aging effect. We try to model that aging effect, which is usually it should be a very slow effect. And we can see that how this slow long-term effects can contribute to this correlation structure also. This is basically the goal of this study. So in order to do this, so we analyze two longitudinal data sets. So one data set is a Simon data set, which is very unique because it is one single subject that was scanned for over 70 sessions over the span of 17 years. So one subject. So it's really rare that there's like one subject like cover this very long age range. It is very useful because we can model some age effect because if we want to model the age effect, we need a large age range. But we know that because of the technical development, usually it's very difficult to find very long-term scans. But this one is unique. But of course, the limitation here is that the data was scanned in different scanners and likely using different parameters. But by some kind of a standardization method, we can still analyze data to get some sense of how these different effects contribute to the correlation structures. And the second data set is like I think all of you have already familiar with, which is the Alzheimer's disease neuroimaging initiative, which is a longitudinal data set that has PET and structural MRI and also functional MRI. In this case, we only analyze the healthy subjects. So we only have and also we want to have this longitudinal data. So we identify data that have at least 5 sessions. So we have in total 72 subjects that have FDG PET data and CC5 for that have at least 5 structural MRI data, and also only a subset of data of subjects have a functional MRI data, resting state functional MRI data, and there's only one run. So we simply average them, have only one average of resting state connectivity matrices, and basically we want to study like how different correlation structures are similar or not using different data sets. So the first is the Simon data sets, right? This is the one male that was scanned for 73 sessions in total. We have a structural activated MRI, so we use segmentation, and then we have this green matter volume images. And for unfortunately, there's no PET data here, but for fMRI, we can still measure something, which I think is kind of a approximate to PET somehow. Is that we can use it is called regional homogeneity. Basically, it's measure if you calculate only one 3D image that result from this resting state scan. It calculates the correlation between a given voxel with the neighboring voxels. right? It's how locally these regions are correlated with each other. They will have a map for one subject, which is kind of, you can think about this similar to a FDG pad image. And of course, we have also the resting state fMRI time series. We can calculate the correlation within the runs. And then those matrices, we can average cross subject, cross different sessions. We have one average functional MRI correlation matrices. So we can compare the correlation matrices between these three conditions. And we use the brain population. We use 100 critical regions from Schaeffer's atlas, and also subcritical regions. We use AAL, 14 subcritical regions, including basal ganglia, amygdala, and hippocampus, and so on, and thalamus. And the matrix, when you look at the matrix, it will be the regions is organized in this way. There are 7 networks, right, from the top to the here to the 50s. There are left hemisphere, and then there are right hemisphere from 51 to 100, and then there are subcritical regions at the end. So this, you will see if there are highly correlation within subject within the different functional modules, you will see that kind of square-like structures along the diagonals, and also left and right corresponding regions along diagonals here. So first of all, we show the correlation matrices, right? We see this resting state functional MRI correlations average over all the sessions, and we do see that kind of structure, right? There are like square-like structures along the diagonals, and also from left and right corresponding regions. And we also see this cross. This is within one subject cross 70 runs. There are correlations of gray matter volumes, and also correlations of regional homogeneity, right? You see that they are not looks exactly the same, right? Of course, you see that there are differences, but fundamental ReHo, you see that there are some kind of structures very similar to this functional MRI, right? And then if you calculate the correlations of the matrices, the correlation between the functional MRI and the ReHo has a higher correlation. It's correlated 0.39. Even this gray matter volume, they does not really look exactly the same, but you still see the correlation. Correlation is about 0.29. So this correlation is already very high, or at least correlate similar to what we reported earlier, that between the FDG pad and the functional MRI, right? So you see this structure, like there are some kind of structures in the here. Fundamentally, you see this region, right? They are within the modules, but also you see that kind of a big structure. It's like all the prefrontal regions maybe, and all prefrontal and temporal regions may be correlated, right? But still, if you calculate correlation or like as a whole, you see that correlations. But of course, the correlation is not that high. So I think the main point here is that the effect size is very important. This one is like higher, but this one, I mean, they are you always see some kind of some level of correlations, but it's not very high enough, in my opinion. And then the question is that how to understand, how to interpret this correlation of the gray matter volume and ReHo, right? So that is that I tend to want to attribute this to, because this is one subject, there's no individual differences, and over the time of between like 20s, some 30s to 40s, a large range, it is expected that maybe there are some kind of gray matter volume changes, right? And that changes, whether that change can be, you know, how that may contribute to this correlation structure. So well, first we plot this gray matter volumes region for all the regions, right? Each line represents one regions, and we have different sessions here, right? You see this. For illustration purpose, we just transformed it so that you can see the overall trend for across different regions, and you can see that, and you can imagine, right? These are like from 30s to 40 something, and then you usually the steadily gray matter volume usually will be just decreasing in this linearly, right? But also some of the regions maybe first go up a little bit and then going down like that. But because we have to somehow standardize this measurement so that we divide it by the global signal, so that maybe they will be normalized, so that maybe their trend is like first going up and then going down, or something like linear changes. So, and I think that is only if we consider this age effect, right, that is the only explanation, and that we can measure this or we can model it using some kind of smooth trend. There cannot be, or I really don't believe that there will be some kind of very sharp differences going up and down fluctuations over this short period of time. So we can model this by using cubic age effects here, and you can see that some certain regions will be going down and then going up a little bit, and a lot of other regions will going up and then going down like this. And that is only component I believe that if there is something that contribute to this correlation matrices, its age effect is like this. And of course, then there will be residuals, right? And this residual, I interpret this as some kind of pure noise. But I mean, I don't know how otherwise you can interpret this. But anyway, then you can calculate the correlation matrices for these two, for these three data separately, right? And this is the raw data we have shown you before in the previous slides. And then the cubic model, you can see that if you show similar to this one, maybe a little bit, but you can imagine that if it is a slow effect, right? This kind of trend cannot be differentiated. There are not many different modes, right? Of either it's going down or going down first and going up, or going up first and going down. If those are true only like trends, if you calculate correlations, the correlation could be either very high or very low. You can see that those correlations are either very red, close to 1, or either very low, which is close. It's very blue is close to -1, something like this, right? This is structure. And also there are residuals like this. So you see that this cubic age effect, if you see these two correlations, these correlations have a very high correlation. It's 0.8 something. So it kind of maybe it's driven by that kind of it can be one explanation is that the age effect, slow age effect can contribute to this correlations within subject correlation of the gray matter volume correlations. But I think one thing that bothered me here is that even the residuals, you see that kind of structure. And this structure, it looks more similar to this raw data. And this residual and this and the raw structure have higher correlation. The correlation is about 0.9, 0.91, even higher. So for here, I don't really know how to exactly interpret this, but I think one of the issue here is that those different data, you see this kind of a fluctuation here, right? Those fluctuations is likely, I would like to attribute this to, the differences in scanning parameters, for example, all the, the scanner, in-scanner inhomogeneity of the magnetic field in the scanner, right? In that case, that may generate some kind of more systematic effect, similarities. For example, a lot of different directions, right? X or Y or Z inside scanner. And that may result in, systematic changes in estimates of the gray matter volumes. That is kind of a geometric changes, right? Maybe can also give you some sort of correlation structures. If that is true, right? Basically, I think that we need to think about these correlations. Maybe that will, if that can give you some kind of correlations, right? That maybe is more. When you think about what is the null hypothesis here, right? We calculate correlation of matrices. We always find some kind of correlations, and here we show some correlation about 0.2 or 0.29. It is not very, it's not very big, but still it's significant. And then that levels of correlation may be simply due to noise or the different variation in scanners. That is one of the issues here. And then we are talking about the resting state ReHo here. We are doing the similar thing that we have 114 RIs here. We show this all the time points. Again, we can model this by using a cubic age model. And you see that whether there are decrease or increase in that two different modes. And also here, there are residuals. Here, the residual, I would like to say, okay, this residual maybe there are still noises, but this maybe there are some kind of components of useful information because the neural activity, they reflect the neural activity, right? If the subject are thinking about different things, right, the certain region may be showing different activations. So that can give rise to this, even though like maybe within weeks, that value may change, right, over time. So so we have the age effect and residuals, and we can also calculate the correlation matrices, right? We have the raw matrices, we have the cubic age effect matrices and residuals. The cubic age effect, you see it similarly, right? Because if we have this long-term effect, it can only give you either very high correlation or very low correlation. You see very reddish or very bluish. But the residual here, this one is, I like this one because you see that this really shows, this one have the correlation of 0.99 with the raw data. Basically, this show you, okay, ReHo, maybe they are reflecting some kind of neural activity, even though after you removing out this long-term effect, that state-like components can give rise to the correlations of the ReHo matrices. It is like this. So here, I think the ReHo, it shows that there are some kind of component of state-like. For GM incremental volumes, that part I tend to attribute that to noise. So you see that there are differences in the functional and the structural correlation matrices. So just a little bit discussion here. I think that it illustrates that ReHo correlations were mainly driven by this state-like neural activity, which is what we want to have. But long-term age, also we show that, I think that this is a unique data that we can show that there's long-term age effects, how this long-term age effect can contribute to the correlation structures. It can give rise to a correlation structure that somehow correlated with functional connectivity matrices. However, the problem is that this residual is also correlated. So maybe I think that simply some kind of noise technical issues can also give you a correlation, a correlation between the correlation matrices. So that's basically is the first analysis, and tell us that when we calculate the correlations, it's not only statistical significance, we also need to consider the effect size, whether it's only a small part, maybe it's only due to noise, and also we need to consider what is the appropriate non-model for the correlations of matrices. This is the first part, and the second part basically it's not entirely new here because I have reported some of this part of this analysis. So we have using this ADNI data. The good thing we can replicate some of the analysis of our previous study. The good thing for this one is that we have a relatively large sample size, right? We have 70 some subjects that have FDG pad and 60 some subjects have structural MRI data. The good thing that there are some longitudinal data. There are at least 5 sessions. But of course, when we want to calculate this correlations, intra-subject correlations, we only have 5 to 9 time points. Then that will make this correlation matrices very noisy, right? But our strategy is that for each subject, when we calculate intra-individual correlations, we have 5 to 9 sessions. We can calculate one's correlation matrices for each subject, and then we average them, right? The average, we hope that the average will reduce the noise so that we can increase the signal to noise ratio, and then the results image will be more clean. And at the same time, we can also calculate this within subjects cross subject correlation. So in that case, right, as we already showed that there are age effects, long-term age effects. Here we want to control for that. So what we did is that, for example, we just use all the data from the exactly same age, biological age, right? For example, 80s. We find all the data across different subjects that have exactly the same age. Then we calculate the correlation coefficients, and then also we have different age range. We can average them together. We have only one averaged inter-subject correlation matrices, and we have it for FDG pad, and also we have it for structural MRI or gray matter volume data. For functional MRI, we only have 17 subjects. Each subject have only 1 runs. We calculate this resting state functional connectivity for each of the 17 subjects, and then we average them. Then we only have one averaged functional connectivity matrix. Again, we want to see how this different correlation matrices are related to each other. First, we look at the structural data, right? We have this resonance function MRI. If you remember the previous similar data sets, this correlation matrices looks very similar to those, right? Even though they are from different samples or using different scanning parameters, this resting state function connectivity matrices is always very similar. And also you see that this structural structural correlations, this is also, you see that like along the diagonal, there are some kind of square-like, right? Also between left and right hemisphere. Right, this looks cleaner than the similar data set. And also these two, you see that they are highly correlated. And here we are mostly interested in the correlation to see whether this structural coherence, whether this is correlated with the function MRI, right? And we show that they are correlated, and this correlation is a little bit higher. I think it can be attributed to that we have a larger sample size, we can average them together, so the data is cleaner. And you can see that the correlation between this gray matter coherence, gray matter correlations, either intra-individual or inter-individual, you see the correlation is higher with the resting state connectivity. The correlation is about 0.4 something or 0.37. They both are little bit higher, right? It is considered moderate. And of course, and then we have metabolic FDG PET inter-subject and intra-subject correlations, and we can also correlate this with the resting state connectivity, and those correlations are even much higher, right? And you see that there's this structure, square-like structure here. But it's very similar to resting state connectivity, and then the correlation is about 0.71 or 0.64. Either it's an intra-individual or inter-individual. So this correlation is like, I think it's the highest among the literature, certainly higher than the paper we published in 2017, right? We have a correlation about 0.31. So I think one reason is that we use more subjects and then we average, right? And also maybe it's in the pre-processing steps, we have improve the preprocessing. So all of those can result in a higher correlation between this matrices. I think this is very important because if you have a correlation about like 0.71, it's like it's explained like about 50% of variance. It's considered a very large effect size, right? It's not the same as like 0.2 or something. So we also show that within each of the modality, right? Either it is gray matter volume or FDG PET, we show that inter-individual or intra-individual correlations are highly similar with each other, right? And also, especially for PET, we have a correlation about 0.7. So that kind of I think it kind of validates the usage of these methods to study functional connectivity, which is already very similar to the resting state functional connectivity using fMRI. And these high correlations, right? I think the question is that if it is due to improve the quality and the data processing steps, right? And then how much can we improve later, right? There must be some kind of limit there. And also, if that is the case, then it means that when we measure the data, we need to improve the signal to noise ratio. We need to improve the data processing steps so that we can improve the correlation. Or otherwise, if we only have a correlation about 0.3 or so on, it is quite limited in my opinion. So that is the take-home message here. And of course, because we have these two data sets, we sometimes we only focus on like certain correlations between two matrices. But if we step a little bit back, right, and then we look at the whole pictures, right, we have all the matrices. If we just examine out the correlations of all these matrices, and then we can see the whole picture and see whether what are the overall patterns of these correlations, which region, which modality will have a higher correlation, and which modality will have the lowest correlation. And also, we hypothesize that if different modalities have used the specific information of that modality, in that case, then from the two different data sets, the matrices from the same modality may have higher correlations. That is the hypothesis. So therefore, we combine all the matrices together. We have a similar data set. We have 3 matrices, right, from resonance fMRI, and also gray matter volume correlations and the ReHo correlations. They are all within subject, one single subject. And for adenine, we also have fMRI time series, and then we have intra-individual and inter-individual correlation of gray matter volume, and intra-individual and inter-individual of PET correlations. We have all together, we have 8 matrices, and then we can show the relationship, the overall relationships. And if we plotted the overall correlations, right, then you can see that all of them are reddish, which means that they are all somehow positively correlated. And here the effect size matters, right? And then you see that the lowest correlation are between, for example, the functional MRI to the structure data, even though they are correlated, right? The correlation statistically significant 0.2 or something, but you see that those are between structure data and functional MRI data. So basically, they are correlated, but it's okay. They are only okay correlations. But of course, here we are more interested in this part of the matrices, the upper red part, which represent the correlation between the two between the two data sets, right? And we are interested in, for example, we have this functional MRI and the gray matter volume and the ReHo correlations, and we want to find the highest correlation corresponding matrices in adenine data, whether they are from the same modality. And the results actually show that. For example, the fMRI data correlation from Simon has the highest correlation with the fMRI data of the adenine. That high correlation is about 0.7 something. This is a very high correlation. And even for gray matter volumes, right? The gray matter volumes from a Simon data set also have the highest correlation with intra correlation of the adenine data, right? That correlation, yeah, 0.4 something. It's still a very big, a moderate correlation, 0.4 something. And then for ReHo, right? The ReHo, even though it is not exactly the same as the PET, FDG PET measurement, the correlation, highest correlation are still the last two, right? The correlation with PET correlation, intersubject or intra-subject correlations or PET from adenine. So basically, this shows that there are some kind of correspondence within modalities, right? But for example, these gray matter volumes, they may reflect similar kind of age effect. I don't know what it is, but maybe age effect that give rise to these correlation structures in both Simon data set and also the adenine data set. The correlation is pretty decent, 0.46, right? And of course, because all of those correlation matrices are very similar, right? That we can perform like a principal component analysis, and we can see, for example, the first component can explain 59% of the variance, and we can show that the first component of this correlation matrices, and this is basically what we usually expected, right? They are within subjects, within module correlations, square leg, and also between white matter, between left and right hemisphere, and also within the subcortical region, you can see that there are, whether one is basic ganglia, another one is thalamus, and so on. Those kind of structures. And if we plot the load of the PCs in the first component and second component in this very low dimensional space, and you can see that in different types of the matrices, they are distributed in this way, and you can see very clearly they are separated, right? And then you can basically see two clusters. At the bottom left, they are all gray matter volume kind of correlation matrices. They are grouped together, and on the top right, they are all related to functional, right? They are resting state connectivity, and also the PET connectivity, either intra and inter. They are all look like that. So basically, it shows that sometimes we want to see whether we are struggling with, okay, whether this gray matter volume is correlated with functional MRI. They do, right? But if we look at the whole picture, they have the smallest correlations, and the highest correlation are among all of those functional measures of connectivity. Even, yeah, for the PET, right, they are more correlated with functional MRI. But even for the ReHo correlations, they are still correlated with PET intersubject correlations. Okay, so these are all the analysis we have. So a little bit discussion about this part. So we see that there are highest and lowest, right, part. The PET correlations are the highest among all the correlations, which first further support like we can use that functional PET data to study connectivity. And also here we show that there are modality specific information, right? Either functional MRI or cross individual functional measurements. Those measurements, if you use different measures like ReHo or PET, they may reflect similar kind of underlying neural activity and so on. And also, despite there are statistically significant gray matter volume correlation had the highest, lowest similarity with functional measures. So these are some kind of a thing that we need to consider if we really want to study gray matter volumetric correlations. Okay. So just general discussion here. So just by using this longitudinal data, do we try to differentiate, at least we just to illustrate how different components, inter and individual variability, and also long-term effects may contribute to the correlation matrices. And we do show that inter and intra correlations of functional measures for PET are highly correlated with resting state connectivity. To me, I think it's kind of a validation of using those methods to study functional connectivity. And of course, here the issue is that previous studies shows lower effect size. Here the effect size matters. In order to validate, we need to make sure that this correlation, this effect size is big enough, right? So in order to achieve that, maybe we need, for example, we have a larger sample size, we have a better data quality, and also better data processing, so that we can ensure that this correlation matrix will have higher correlations with measurements such as functional connectivity. And also here we show that, for example, ReHo and also PET, there are some state-like components that may contribute to these correlation matrices, which is good, right? But it's not clear how this neural activity, for example, during the scanning time, 5 minutes during the resting state, or if we have maybe half an hour to an hour of a PET scan, how this average times like related to neural activity is related to, for example, moment-to-moment neural activity as measured by functional MRI. How that neural activity are related in different timescales are still lacking studies. I think that part needs us to study in order to fully validate how to explain the relationship between, it's a very static measures of brain function to neural activity. And also we show that there are long-term age effects here. We just show what it will look like if we want to attribute that correlations to long-term age effects. But it shows that there are some, if that's the case, it will show very high correlations, right? Either very positive or very negative correlations. So that means that there is a kind of a consideration. If you want to really consider, okay, if you want to interpret this age effect as the main drivers, you may need to, for example, you model this age effect like that. Even though you are using cross-section data, you can still model the age effect, right? And of course, to improve the interpretability, maybe we also need to consider, for example, remove age effects, or for example, we restrict the age effect so that we have similar age, for example, that will minimize the effect, this kind of effects. And of course, the results suggest that the effect size really matters. All the correlation are significant, but the correlation of 0.2 is very similar, different from a correlation of 0.7. So that effect size matters. And because of the correlations like gray matter volumes, I think we need to consider what is the appropriate non-hypothesis for this correlation of matrices. Lastly, we just have some limitations I want to discuss. First of all, of course, we use these two public data sets, which is unique, but it is have some sample bias, right? The age, the ANI data are all very old subjects. This single data set is only one individuals have the age at the age of the middle age range. So we need to test this in the larger sample size, for example. But I think that the data is really difficult to acquire, to collect, especially for like this kind of single data set have a very long age rate, like time span. And of course, there are missing data here. We do not have pet data for the signal data. So, and also for adding data, if we have more longitudinal data points, then we maybe have more reliable measurements or estimates of connectivity. So, and then lastly, I think one of the issue is I only use the residency connectivity. For me, it's more convenience. But I believe that it is more, if they are correlated, they should be correlated more, especially PET, functional PET, right? They should be more correlated with the functional measures of functional MRI. But certainly there are other references, for example, diffusion intensity imaging. There are also studies like from Igor's group, they show that diffusion intensity imaging when metatracked can also explain a lot of variance, but that part we do not have that in the current analysis. But of course, when we discuss like which reference is better, maybe, so I think a lot of audience here may not agree that at all that, okay, the PET is PET, they may have their unique, the correlation structures and so on. So that is also true that maybe when we say validate, it's not like we want to correlate it with the functional MRI measurements. Maybe, I don't know what is the reference for that point, but anyway, so maybe PET have their unique correlation structures that maybe we do not really correlation with others is good, but it's not necessarily the golden standard. Okay, thank you very much for all attending here. I will send all the lab members and collaborators from especially Christina. This study, we have a preprint. If you are interested in the details, you can scan the QR code. And also, I would like to thank all the open access, right? It's a data set from the SIMO data set and other data sets. Those open science efforts make this data analysis possible. And also, we send fundings from NIH and also the autism grant from NJ government. Thank you very much.
Thanks a lot, Sing. Let's congratulate for this great talk. I think it's a statement of facts. We could summarize what you show here. I'm not going to waste too much time talking. I'm open this seminar to questions, and I know Chris had already a few already in the chat. I don't know, Chris, if you want to shoot your questions here.
Yeah, I had quite a few questions. I didn't want to interrupt at the time, but maybe I should have done it. I was sometimes a little confused whether in the intra-individual matrices, whether you were showing just one example for one subject or these were averages. But anyways, I mean, that's maybe for annotation later. I would actually be interested in going to the Simon data set at the end, the last slides you showed where you did these age, yeah, for instance here. Yeah, I was just one clarification for me. So the bottom row correlations, by the way, I think this is a very good idea. The bottom row correlation, these are across individuals, correct? Or did I get this?
Cross section, this is only one individual cross sections.
Oh, so this is one individual, but somehow you applied the age model and still got residual sort of values in each ROI.
Yes. So basically,
but across different runs of the same individual. Oh.
Right. Basically, you see those lines. Yeah. Each of the lines, their correlation will give you this correlation matrices. The residuals also, these different lines give you a correlation matrix like this.
Oh, I see. So in other words, I find one individual and I see, and then for all different regions. Okay. Okay, let me. I have to digest this.
Are you satisfied, Chris, with the answer?
No, I am. Now I have to think about.
Okay. I let you to thinking. Is there anyone with other questions? Otherwise, I have one for you, Xing. It's
Right.
like recently, if you read the literature, there is the coupling uncoupling business in MRI. The structure and functions are coupled, locally. So why majority of the analysis that we do here, they're taking into account the entire brain at the same time. So how do you think we can adapt these methods to study more regional coupling or similarity between modality?
Yes, I don't know. So I think a lot of studies I saw is just like a correlation between structure and the function, right? They are kind of coupling. It's interpreted as a coupling. If I understand your question correctly, there are some studies just use. Is it still whole brain? I think the structural measurement and the functional measurement, they are correlated. They interpret this as a coupling. Is that correct?
I mean, they look more at individual structures, and there are regions where the fMRI signal is coupled with the underlying structures,
Okay.
depending on the if you want, the functionality of the brain. So I was wondering whether we could more study this similarity between modality more on a local level, if that could give us some better insights.
Yeah,
Or
it could be, but that part I'm not familiar with. That need a finer spatial resolution, I think. But here, yeah, we focus on like very whole brain thing. Locally, I agree with you, but I'm not quite familiar with the literature.
Okay.
I think that is also.
Yeah, I mean, not to, sorry, I don't want to butt in, but I think it would be fairly easy to study that. I mean, you could either take the nodal strength where you just look at correlations or connectivity to the rest of the brain of one area, or you shrink it down to a kind of regional homogeneity measure, right? But in different modalities and related. So I don't think it would be that difficult, but anyway, it's my biased opinion.
So I'm aware of time that we are running a little bit late. So before we continue with discussions, I have two news. For one is like, please leave your feedback about this talk. It's very important for us to organize future meeting. And do not forget to register to the next seminar, be in April, where I'm going to present together with Mario Severino, recent application molecular connectivity on neurotransmissions. In the chat, you have all the links Arianna just shared, so please do not forget to put your contribution to this. I'm continuing with the discussions. If there is any other questions from the audience, I see many friends in the audience, so I'm sure there are questions. Where is Friday? Arianna, do you have one?
I have a question. So, I mean, I really like your slides where you show like the decomposition of inter-subject connectivity into neural activity, age effects, and everything else. So the question for me, okay, let's say that this is very accurate model of how to, individual variability and connectivity are influenced and divided. But then if I have an inter-subject connectivity matrix, how do you think there is a way to model the individual effects, and say this is related to this thing and this is related to the other thing? Because that would be really great, right?
Yeah, that would be great. I feel that difficult task. Yeah, here I just want to show, okay, if we can control how that single effect can make effect. But yes, if you have cross individual data, I feel it's difficult. I don't know how to do that. If we kind of either control some of the effects or no, yeah, I think that is very difficult. Yeah.
You think is it theoretically possible?
I think theoretically still, I think if we can do within subjects measurement like the FPET, right, it will be better. Or we have multiple measures, multiple session measures, maybe also. Yeah.
Okay, something to look into.
Thank you, Arianna. Any other question from the audience?
So I have one question.
Yes, please go ahead.
So, Dr. D, so here noise is quantified as a residual after modeling.
Right.
Is it so? So, I mean, the other factors, I mean, maybe may have the impact in the prediction, right? So the noise is maybe I mean whether the noise is the exact term that it is contributing to the individual variability, maybe individual differences if you consider, then that the gender things also may come, right? So
Yeah. So here, I think the model is simplified. Fundamental gender, sex, and other factors. Yeah, it's not exclusive, I think. I agree, of course, but the noise is, I mean, just theoretically put it here, right? Certainly there are other factors, I think.
okay.
Thank you for the question. We have another last question from Vesna. Would the PLS analysis possibly help to investigate dependencies? Partially squared, I believe she's referring to.
Oh, yes, I think that would be, but yeah, we have done something like lasso some before. Yeah, I think that partial least square is, but we haven't done that. Of course, multivariate analysis is something we can do, but yes, here, but we only focus on the correlation matrices, so we did not do that. But I agree with her.
I mean, our results from our lab really show that through PLS you can distinguish few factors. Could be age, could be affinity. We're talking about TSPO. Even scanners probably are adding to the elements. If you have a subject acquire different scanners, when you have a multi-site data set, are also factors that will contribute to this model. So the problem is that when they become collinear, and then then you are limited by the theoretical limited by the fact that you cannot distinguish two completely collinear effects. But definitely these are good methodologies to
Yeah, I mean, so sorry, I mean, with PLS, I'm not sure. I think PLS is a great method, right? But you'll get latent variables that are given loadings in terms of all these things, age, scanner or whatever. So it's good for that. But if you then want to capture things that are orthogonal to these things, that's not going to be loading onto anything, right? But we're also interested in these irreducible things that are not explained by age and all the other things, right? So then I think maybe some of this regression across the group of individual nodal strengths might be. I mean, the two are complementary, ideally. You use them both.
Thanks, Chris. That's a very good remark. I guess if there is no other questions, shall we close the seminar here? I'm aware of time. Then you don't want to say that Italian moderators keep the meeting too long. Thanks a lot.
It's entirely your fault, Mattia.
I know. I take all the blame. I wish everyone a very happy Friday afternoon, a nice evening. Thanks a lot, Xin, for the fantastic talk, and see you soon at the